I think it is clear to believe in the power of ideas.
Fresh thinking of the Manhattan Institute.
Thanks again for joining us at the conference this morning. We're going to start with our second panel now, which I'm going to be moderating. Everyone has bios in their packet, so I'm not going to introduce our panelists this morning at any depth, but let me just say, to my right is Linda Avey. She is the founder and CEO of Curious Inc. and the co-founder of 23andMe. Next to her is John Crowley, the CEO and president of Amicus Therapeutics. Next to him is Karen Gutekunst, who is the Vice President for R&D at Clariant, which is a subsidiary of GE. And then finally, we have Andy Palmer, who is a serial entrepreneur and venture capitalist, and is the founder of Koa Labs and the former president of the big data firm Vertica Systems. So the purpose of our panel, following up on Peter's comments this morning, is talk about the technologies from the ground up. So Peter talked about where the science is going, the challenges it's meeting in terms of FDA regulation. But to my right are seated people who are in the trenches every day working to advance patients' access to breakthrough therapies and really doing things that promote disruptive innovation in the drug development space. And I'd like to talk about those disruptive innovations. And those kinds of technologies that I have in mind are electronic health records that have the potential to bring together what we call genotype data, so genomic data or other omics data, phenotypic data, maybe that's clinical outcomes, and other data, along with social media platforms like Patients Like Me or 23andMe. And feed that information from patients back up to researchers, whether it's at the NIH or at drug companies or at the FDA, and then the tremendous potential of a whole suite of molecular diagnostics that Peter touched on earlier that are rapidly dropping in cost and increasing in their predictive power. And then finally, these enormous big data tools that are widely used in other industries like retailing. You probably have never had an interaction with Walmart that wasn't captured in some way and that it feeds back into its system to improve its delivery, its ability to bring new products and price products for consumers. Those tools are just beginning to be felt in the healthcare sector, but can be transformed. So my first question to the panel here, and I'll ask them to answer it for about 3 to 5 minutes and then lead a panel discussion, is how are some of these technologies revolutionizing drug development and helping us improve patient outcomes? And in particular, what do regulators and companies need to do today to leverage the full potential of those technologies to drive rapid patient-centered innovation? And I'll just go from right down the line. If Linda, you could start.
Yeah, thanks a lot, Paul, and it's great to be here. I really enjoyed Peter's presentation, and I guess my perspective on this is I'm always looking 5 or 10 years out to see, where are we going to go from cancer? We've made a lot of progress in cancer, and one of the reasons, of course, is because we have a tumor to interrogate and apply these technologies to generate the molecular profiles that are so useful and that will be part of adaptive clinical studies, hopefully very soon. The problem is then when we start to move into neurology and other spaces where we don't have a tumor, and how are we going to approach these diseases and generate the data that we'll need to continue the progress we've made in cancer. And so my perspective on it is that getting patients involved directly. One of the other issues, of course, is that a lot of neurologic and other diseases are multifactorial and probably have numerous, if not in the 10s and 20s of subtypes. And how do we start to break those all out? So getting patients directly involved and then starting to employ the technologies that we're starting to see now coming out in the consumer space where we can start to wear sensors and devices that really track very interesting data. And I'm not talking about Fitbits and Nike Fuel Bands. I'm talking about things that we'll be able to wear that will pick up our electrochemical makeup and how that's fluctuating throughout the day, our core body temperature, glucose levels, all kinds of data that patients will be willing, and I should say people will be willing to wear. And then how do we start to feed that information into drug discovery and generating better targets for discovery, but at the same time then having the genetic profile in the backdrop as metadata that we can look at and see what profile people have as far as their liver enzymes go, and so incorporate that information in as well. So putting the patient at the middle of all the data we think is the right approach, and then giving them channels to share it with others, whether it's their favorite researcher at Memorial Sloan Kettering or someone else at another institute, or maybe at all institutes. Maybe people want to share their data out as much as possible. But I really think that this is the right strategy as far as going into drug approval, that we let the patients have more of a voice.
Great. So I work in the field of rare, orphan, and genetic disorders, and in that field actually wear two different hats. I'm an entrepreneur and have been for almost 15 years, helping to start companies involved in developing medicines for these many of these disorders. But I got into it originally as the father of two young children with a rare neuromuscular disease. And when you think about technologies, for the better part of those 15 years, I had been focused on technologies that would advance medicine, that would provide new medicines, treatments, cures. But more and more now we're trying to understand that even within that focus of advancing medicines, and the prior discussion, Paul, was absolutely spot on in what do we do to accelerate medical research through that enormous bottleneck of drug development, obviously with the FDA as the gatekeeper. For me, a good part of it is how can we apply these technologies, especially patient-centric technologies, and all the information literally down to a patient-by-patient case study, as you look at each of these diseases, to help us design better clinical studies, to work with regulators, to use a lot of these new tools that are available in some of the legislation that's just been passed. So previously, people had, you'd get a drug approved, and then you'd work with patient groups to provide access, to provide reimbursement. In the rare diseases, often to build patient registries, to collect data. We're trying to do that more and more before we even get into clinical studies. If you step back and you look at some of the rare disease drug development, one of the very first drugs developed for a rare disease was an enzyme replacement therapy called Ceridase for a rare disease called Gaucher disease. Affected what was thought to be a couple of hundred people, and in fact it ended up being several thousand people. The first clinical study, very creative, adaptive, 10 patients, enzyme replacement study given largely to children, and it failed in 9 of 10. And then they went back to the FDA and proposed, on the heels of that great success, a phase 1, 2, 3 combined study in a much larger number of 12 patients. And this was when they were actually developing the drug and making it from purified human placental tissue. And this was in the late 80s when HIV was rampant in the blood supply, so enormous risk. And the FDA, much to their credit, I think a lot of this pressure actually came in the times of the HIV activism, had to apply a very different framework, the risk-benefit analysis that hopefully they're starting to get back to. But there was very limited data about how each of these patients may or may not respond. That drug went on to be approved with that successful study. Other drugs for rare diseases like Fabry disease, a disease that my current company is exploring with the next generation of a enzyme replacement therapy. But as medical science advances, we're trying to catch regulatory science up, but caught in the middle is what do we do with all this data on a patient-by-patient basis? We used to understand Fabry disease as a rare genetic disease, X-linked, so thought to affect symptomatically at least only males. Approved the first drug 10 years ago this month, Fabrazyme, and it was thought to be a very homogeneous population. Turns out that females are just as symptomatic, even though they weren't studied in the original pivotal study. Children are as symptomatic as adults. But, we are all polymorphisms, and as we know, even people with rare diseases have a great amount of variability in how they respond to medicine. So a lot of the technologies that we're looking at, Paul, are trying to understand going into the clinic literally on a mutation by mutation basis. And Fabry is a great example because here's a disease that affects several thousand people, potentially even many more, that's caused by over now 800 known mutations. So you're going to get down to, and we're already doing studies that incorporate many of these mutations where people are going to respond differently to drugs, and our ability to use these technology tools to capture really what is the essence of the patient population on a patient-by-patient basis, by genotype, by phenotype, and to design those clinical studies, I think is going to have the greatest impact, at least for us in medicine. So that's where we see technology playing such a great role in capturing that vast amount of data.
Thanks. As Paul mentioned, I work for a laboratory called Clariant out in Southern California, and we perform testing on cancer patients every day. So we use the tools that are that everyone's talking about, molecular profiling of tumors, you know, whether it's immunohistochemistry or FISH or PCR or DNA sequencing. That's what we do on tumor samples every day. And if you're not aware, you should be aware, this is also a very highly regulated industry. It's not just the drug industry that's highly regulated, but the device industry is as well. And the laboratory industry is becoming more and more regulated. So the technology that we have available to us today is phenomenal, and it provides us with so much information that, you know, I think patients are not at the center of what we do. That, you know, we're sort of technology driven and we understand what's happening, what's driving cancer. That information is overwhelming to physicians. The community physician is, you know, I mean, Memorial Sloan Kettering, of course, is doing great work, and they understand how to utilize molecular profiling of tumors. But you can imagine the community physician having, you know, a patient report that has 100 or 200 genes describing 50, 60 mutations, and what is that physician supposed to tell the patient to do with that information? It's really complex. On the regulation side, you know, I think I can count on one hand the number of true companion diagnostic tests that are approved today. And those labels of those tests are extremely limited in the amount of information that can be reported by those. For example, the EGFR test that was just approved, I believe maybe yesterday or the day before by FDA, it reports a limited number of mutations. There are, you know, over 20 variants that are known in 4 exons of the EGFR gene. Those are not all reported in that test that was just cleared by FDA, even though the technology is capable of doing that. And that's because the statistical power of the clinical study that was used for the clearance of that test didn't have enough patients with each one of those different variants to allow the inclusion of those variants in the label. So I don't want to bash the FDA, but I believe that in the EU, for example, there is greater access to more state-of-the-art technology more rapidly than there is in the US, which is not putting our patients here in this country at the center of this discussion.
Great. I just like to start by saying that I think that the drug discovery and development business is fundamentally an information business. And the wet lab was always going to be important, but at the core, that we're talking about information and knowledge being generated and shared. And I believe the state that we're currently in is that we have vast inefficiencies in terms of information access and availability. And there are many different examples of this. One of the examples is the average doc trying to consume all kinds of genotypic information or information that's available to them. We haven't made it easy for them. We need a kind of all the systems that doctors use on a day-to-day basis need sort of a complete overhaul, so that they can consume information more effectively. But with all this talk about big data, I think that and the potential of big data to impact the process, there's a real risk that we lose sight of the fundamental problem. So I was most recently running software and data engineering over at NIBR, Novartis Institute for Biomedical Research. And there was, we spend $200-300 million a year working on building tools for scientists that help them, do all the great work we do in rare disease and other indications. And there was an experience that we had that kind of changed our perspective. We had a call that came in, random call from a patient that had been diagnosed with a rare form of cancer over in the UK. She had talked to her clinician. Clinician gave her maybe 6 or 9 months. She said she wanted to do whatever she possibly could in order to help, save herself, and but even if that wasn't going to happen, to help someone else. So she goes on the web. She goes to all the web resources, the trial resources that we're all used to using, and tries and comes up with a clinical trial that she thinks might be appropriate for her. Can't get a hold of anyone that's involved in the trial, doesn't have any information online, knows that it's a Novartis trial, calls the main switchboard at Novartis, and you can imagine sort of the response that she received, and eventually gets into research over at NIBR up in Cambridge. And again, the switchboard, you can imagine the reception receives, and somehow she gets to myself and one of my colleagues. And we said, well, we just, we don't really know, but let's do some research and let's see if we can figure out how to help you. So we start looking around, we find the information, we get all the approvals. She's in the UK, so we have to go through all these unnatural acts to get these approvals. And we find the trial, we find that we can get her in. We call her back with the name of the primary investigator for her trial, and lo and behold, it's her oncologist. So for me, right, we can spend a lot of time and energy talking about big data and the potential for information technology to revolutionize drug discovery and development. But if we can't, right, bring the right trials to willing patients who want to enroll in trials that help us discover drugs that make a difference in the lives of all patients, then I really think that we're not doing our jobs. There's basic blocking and tackling that needs to be done. This experience at Novartis caused us to come up with a program by which we can take and publish in a machine-readable form all the inclusion-exclusion criteria for all of our trials in an open way that's consistent with the way that all other pharmaceutical companies could publish their trial information, and merely cross-referencing that with people's out-of-the-box EMR records that they already have would make a huge difference and a huge impact. So I'm here as a technologist. I spent my entire career building bright, shiny objects of computer science for the most sophisticated applications in the world. But I really believe that many of the applications, many of the ways that we can make a difference in the world have nothing to do with these bright, shiny objects, have much more to do with helping get the IT people out of the way and just do some systems that make sense for patients.
Thank you very much. If I could just follow up with a couple of quick questions of the panel. I think my first is that the example of AIDS keeps coming back up when we talk about regulatory innovation. John, you raised it, as kind of a high watermark. What exactly should the lessons we take away? And is it also that in a world where, in effect, I think Linda and everyone was addressing this to some extent, we're all orphans in some respect. Like, in some sense, you are unique in some way, or at least you share some molecular profile with some other patients that would make you a member of an orphan disease group if we really knew enough about your molecular profile. So how do we empower enough people, enough consumers, patients, both, to become participants and empowered in this process that would really drive reform in a powerful way? Are there some places we can look to, perhaps based on the AIDS experience, or other things that are happening right now that could be prototypes for what we want?
I would say that I think this is a huge opportunity for foundations to step in. I think a lot of foundations are kind of struggling with what is their purpose, and if they really could, and we're seeing it. I went to the Partnering for Cures meeting here in New York last November, and patients are asking for it, and I think the foundations are responding, and it's a new call to action for them to really embrace their community and then be a voice for them as they go out into the research world and look for funding and look for ways to get the patients involved directly in research. And so I think that's something that we should really explore.
Because that raises patient awareness and empowers them to become advocates for their own disease states.
Yeah, if you look back, Paul, at the HIV lesson, it was in the late 1980s. The initial push was to get AZT approved. And it wasn't because AZT was going to cure all AIDS. It didn't. In fact, it was a horribly toxic molecule. But it pushed the FDA to, when they looked at that risk-benefit, to be more on the side of, well, let's go ahead and approve it and let doctors use it. But what it really did was to open up, especially when they could base it on a surrogate marker, and then, of course, that led to Subpart H and a whole cascade of drugs approved on surrogate markers. That led industry to start to come to the table and said, wait a second, we think there is an accelerated path here, clearly an unmet medical need and a sizable market, but a path that we can understand and we can move forward on. So we tried to copy that in the rare diseases, and the challenge had been, since the accelerated approval based on a surrogate endpoint had been in place in the early 1990s, you had dozens of drugs for HIV/AIDS approved. You had one for any of the 7,000 rare diseases. It was actually Fabrazyme for Fabry. So when we started to look at what we could do to change that, I sat on the board of Bio, and a bunch of us started to look from, especially the small company perspective, what we could do. But we realized we couldn't do anything until we had the patient groups on board. And a lot of us were involved in the rare and orphan disease space, so we went to the rare and orphan community, realized it's great that we've got 7,000 rare diseases with 30 million people affected with rare diseases, but there was no one group to kind of corral all of that energy and enthusiasm. So one of the things we did was put together an umbrella group. Now the Global Genes Project, to go to globalgenes.org and you can look at that, and we've started to brand the community. We got our own ribbon. It's actually made of denim by kids with rare diseases. It's called In Our Genes, so it's a neat little double meaning. But even that wasn't enough, so one of the things we did on top of that a couple of years ago, as this legislation was all coming together, was put together a group called RDLA, Rare Disease Legislative Advocates, where we actually got patient advocacy involved at the legislative level and spent a lot of time on Capitol Hill. And a year and a half ago, when everybody said, PDUFA 5 would be clean and Big Pharma wanted it simple and straightforward, we were able, at least in part, to corral hundreds of rare disease groups to put forward the ideas that became breakthrough therapy and accelerated approval and other pieces of legislation that at least give the FDA another tool set, and certainly a focus from Congress on accelerating research. So, a lot of that lesson came from the HIV community and the AIDS community activism in the 80s. So we'll see how successful we are in the next few years.
In my perspective on the HIV story, I worked for Roche Diagnostics and worked on the first viral load test that was approved by FDA. And the patient community was such a strong lobbying force at the agency to get them to approve the viral load test as a surrogate marker for their disease. And this was a very well-educated group of patients that was lobbying on the government at that time. So, Andy mentioned getting, human-readable information and understandable information to the patients. Linda mentioned putting patients at the center of this information. I think, 23andMe, the story where, people are giving up their own genetic information as part of a central database. These are the kind of tools that we need to enable individuals to become educated and understand what is happening in these very complex diseases so that they can take some ownership of their own health and help industry lobby on FDA, because I think, as industry, we certainly have a very financial incentive to move the agency in a certain direction, but from the patient perspective, it's not being motivated by money, it's being motivated by their health, and I think that will speak more loudly to the agency on these issues.
I'd really like to echo what Karen said. I think that empowering the patient with better information, and the information that exists is incredibly sophisticated, and we learned a lot through the process of patients educating themselves in AIDS. But now with cancer, we're going through a similar kind of a thing as people try and interpret their own genomic information. I think that the work that 23andMe has been doing is really illustrative, right? The interface is very basic. It's very intuitive for the average consumer, and it's also easy to make a lot of bad assumptions based on the information that's presented. And so we have to get much better at figuring out how to make this information consumable by the average consumer, the average patient. It's really, it's a very hard thing to do. But many in the technology industry have spent a lot of time doing this. So we look at financial information as a great reference. 40, 50 years ago, the average person consuming and making their own financial decisions on a day-to-day basis that would determine the health of their retirement income would have seemed like a stretch. Today, a lot of people do that every single day. We need to get to make the same kind of progress in terms of people being able to consume their own health information.
In a phone call we had in preparation for this panel, Andy made a really interesting comment. He said, with the data packages that are out there, everyone can become their own informaticist. So that's a challenge, I think, that you recognize, that in terms of how do you validate data in a way that everyone's going to trust, that the research community is going to trust, that the regulators are going to trust, but also in a way that you package the data and that empowers the patient to become much more of an active participant. And I think last night Linda had said that patients can they're going to go ahead and run their own adaptive clinical trials. They're going patients like me and other people, once the data is in their hands, they will go and chase things down in ways that can be transformative. So do you have any thoughts, Andy, about how you build that trusted network?
I do, and I really believe too that there's a very important role to be played by the FDA and the government as a whole. If this patients are taking control of their own health information, that is going to happen. I believe that this is going to be a liberating exercise. And if we try and control that information too tightly or control the information in a way that's unnatural for the patients, they'll just do it themselves. We need to embrace the fact that they're going to be empowered and provide them guidelines so that they can actually consume this information in a meaningful way, and help provide resources so that when they get into an area of ambiguity or when there are things that they don't understand, that they have a place to go as a resource to be able to interpret this information. And it's, their doctors will probably not be prepared to tackle some of these questions. Patients like me, guys, have kind of validated that that's the case. So we need to empower the docs. But there's a whole set of resources that are going to be required for the average patient to be able to become their own informatician and do these kinds of analytics. We've seen these cases one-off in clinical trials. A lot of the foundations have seen patients doing this themselves. I think it's a very powerful role, to Linda's point, that foundations could become information interpretation and medical interpretation sort of resources for a lot of these patients. I know I've done a bunch of work with the Lance Armstrong Foundation. I've been doing tremendous work in electronic health information for cancer. I think the answer is in there somewhere. But make no mistake, as a technologist, it is not about the actual technical tools. The technical tools exist today, and most of the things that are being implemented, even in state-of-the-art systems, are very, very basic from a computer science standpoint. The resources are out there. What we're lacking right now is the interpretive and information management capability at very large scale across the entire patient population.
Great. With that, why don't we open it up to the floor and take some questions from our audience? Sir, if you could just wait for the microphone.
So I have a question. So Andy, I kind of agree with what you propose, but I sort of disagree with some of those. For example, lots of the data available. So for Linda is interested in neurodegenerative disease. And I think that's encoded in the genomes. And people have spent millions and millions of dollars, government spent millions and millions of dollars on all the GWAS studies. And the GWAS studies just look at the outlier. That means the p-value is 10 minus 27, whatever. So you can only find one gene that could be causative related to that disease. But there are so many interactions we don't know, lower frequency mutations. So that's the big data challenge. For example, these data are there. And the other thing is that in terms of tumor, the TCGA spent, again, millions and millions of dollars. So sequencing is not the answer. Sequencing provides you the genotype. And the eventual output is the phenotype. So the phenotype is the integration of your genotype and your environment. And on top of it, the complexity is tumor is very heterogeneous. We sequence one single tumor, and you can find two different mutations in the same tumor type. So the idea is basically you need to have a standardized and make very good ways of interpreting the data, because the genome, single mutation is not going to tell you what's going on.
I believe we've reached a plateau in terms of sequencing and the value of sequencing. I think that the only way to change the vector that we're on now is by large-scale aggregated data sets of genomic information, and ultimately those data sets being integrated with phenotypic information. But we cannot derive any more value looking at single samples and single analyses of small groups of people. We need a large integrated data set, not only to deal with some of the challenges of quality, but also to be able to run these studies. You can't run studies anymore in these small groups of people and patients.
The GWAS study is not small study. GWAS study is hundreds of thousands of many.
I guess the disagreement is in terms of scale. I believe that we need to get to tens of millions relatively quickly.
But that you can do 100 million people and get the genetic information, it means nothing until you get the phenotypic data and environmental, all the additional data to layer on top of that. And that's been the big downfall, I think, of most of the GWAS, is that I saw another publication, and it came out of Harvard, of a GWAS in autism, and all they do is say, well, we use the diagnostic criteria for autism. Well, that's crazy. It's a spectrum disorder. It's probably 15 different things going on there. And to say that we sequence patients with autism, I get that far into the paper and I just stop reading it. So we need to move on, and we need to collect additional data on top of this. And this is where I think patients have to be directly involved.
It's almost exactly as the TCGA. So sitting on a TCGA, what TCGA told you on the initial paper is, if you have this mutation, your disease is worse than the others. So what? You don't tell a physician how to treat your patients.
Right.
You just tell them you're outlier. That's it.
Sir. And please just state your name and affiliation.
I'm Andrew Reddick from MetaMed. Just as a follow-up for something you were saying, Andy, a minute ago, about how we need to connect the phenotypes to the genotypes. I'm familiar with a mathematician up at Yale who has a relatively novel algorithm, been developed in the last 10 years by her advisor. It's designed to test correlations between things, and she suspects that it could potentially make such a correlation. Actually running it would be fairly simple and ridiculously cheap if she had access to the data set. She's been spent the past year attempting to get access to some sort of data set where she can do this for like tens of dollars, like no money at all. Do you have any thoughts on how something like that could happen? Or I know there was talk about keeping data sets secure, but
yeah. So my experience, we have this project going on at the Broad Institute right now where we're building this large-scale genomics database, and sort of brought a bunch of people together to talk about this, and folks from the Sanger as well as a number of other centers. And it was amazing because all the folks in the U.S. spend their time trying to figure out how you're going to create a rational data set through all the complexity and all the policy and all the consent issues and everything. The folks in the UK from the Sanger, like in the opposite seat, their primary questions were, how are we going to do this phenotypic, genotypic link, and how big is the data set that we need to do it to have statistical significance? What are we, like they're already on it. So my take in getting access to the data in these data sets already exist. They just don't exist in the United States. Now, we in the United States have to decide if we value those data sets and we think they're going to be important to improve human health, and how to create them in such a way that researchers can operate on them, without legal risk. But I think these data sets already exist. They just don't exist here in the United States. I think the best place to go is the UK or Sweden or Finland. Yeah.
And just one other thing to throw in is Sage Bionetworks is assembling a large data set, and they've gotten contribution from several pharma. So that might be a good place to do some testing of the algorithms.
Yeah, Sage is a good starting point. I agree with that. Yeah. John Wilbanks. Yeah.
And Linda, could you just talk about, 23andMe obviously started their own enormous data mining operation. What role do consumer genomics companies continue to play or could potentially play in this space? Because they're collecting both phenotypic and genomic data from their clients.
Well, along the lines of what Peter presented, of assembling large data sets and not just looking at one particular biomarker, looking across the spectrum. And 23andMe is partnering with the pharma industry now to do some data mining to say, okay, we're going to open this up in ways that protect our customers, but their patients, and they have a lot of data to share. So it's very, it's all about collaboration and making sure that, of course, all the protections are in place. But most people, most of the users in 23andMe, if you ask them, they would say, yes, we want this. We don't want these data to just sit here. We want them to be used in useful ways. So that's, I can't speak for the other companies, but that's definitely one of the premises of the company.
Yes.
I'm Gualberto Roaño. I'm with Genomics in Hartford, Connecticut. We're a pharmacogenetics company. I wanted to bring up to the attention of the panel what I call a missing point in regulation, and that is the role of Center for Medicare and Medicaid. Those of us in molecular genetics are going through a crisis right now where the pricing for these molecular diagnostic tests has been thrown to contractors all over the country. Right now we have 13 different prices for epidermal growth factor mutations, and it's a total chaos right now. So the question is, given this regulation that Center for Medicare and Medicaid imposes on innovation, because they pay for it or they don't pay for it or they underpay it, would there be a role of thinking big and integrating the FDA with CMS in such a way that the CMS does not become yet another boundary, another challenge to innovation and patient care access to these technologies? Because right now we have plenty of approved tests by FDA, plenty of drug combinations, and CMS is shutting it down.
And Karen, just to follow up, I think that's a question really for Karen, but Karen, could you also comment on, the role of diagnostics in some of these areas is critical in terms of avoiding serious unwanted side effects, potentially the value of the drug is delivered through the diagnostic, but how much of that value is actually being captured by the market right now in the sense of, what the diagnostic brings to the table?
Yeah, so thank you for raising the topic, because since the panel was only an hour discussion, I didn't want to raise it because I could talk for 3 hours on this topic alone. I think that the way CMS is addressing diagnostic testing, right? They're trying to take costs out of the healthcare system, which I appreciate as a taxpayer. However, I think 2% of spending in healthcare is spent on diagnostic testing, and they're trying to take 60% of the cost out of that. So they're really hitting diagnostic testing hard. And because it's not being paid for, patients aren't getting access to complex molecular profiling of their tumors. You can only get paid for 3 genes or 4 genes right now. You can't get reimbursed for a multi-gene sequencing panel or a whole genome or whole exome sequencing. That is, and the private, the insurance companies follow the lead of CMS. So that's the most you can get paid, is what you get paid by CMS. To Paul's question is how much of the value is being captured. I would say virtually none of the value is being captured, because the way the pricing reimbursement schedules are being determined is on the labor that is in their minds assessed as contributing to the cost. So how much does it cost you to do this test? They don't consider the value of the information that's being provided by the test. It's just a one-for-one, fee-for-service, if you will. And it is really hurting the laboratory industry and will, I think, further drive innovation out of the system, because you'll start to consolidate laboratory testing into the big box labs, right? They're like, we'll have Walmart and Target of the laboratories if things don't change.
And the challenge for, I think, the industry is also, without those diagnostics, your rapid pathway to market or your advantage to be able to prove a survival advantage is just not going to be captured. So that's, is that dialogue going on across industries right now to say, wait a minute, if we don't get the value of these tests captured, the whole, personalized precision medicine edifice is going to, be mired in mud?
I think those conversations are starting. Of course, the diagnostic industry thinks pharma should pay for it if the government won't pay for it, right? Because we're like, hey, look, you know, we're providing you with the information to get patients access to your drugs. And I think that, you know, there are some pharma companies who appreciate that and say, yes, but we have our own problems to solve, right? And they do. They got to get their drugs through the FDA. So I think those conversations are starting, but there is not really a forum for true collaboration across the industry to enable the value to be captured.
Just one comment to that too, from a therapeutic company perspective, as more and more people understand their genome and with specific diseases understand, you know, whether it's a particular mutation or mutations at play like in oncology, we have to be real careful not to reinvent the wheel, because I think the knee-jerk reaction is now, oh, there's a therapeutic, it's targeted to a specific mutation, therefore to market that drug you need a companion diagnostic. In some cases that may be true, but you know, we would argue, for instance, in many of the rare diseases, all patients have already been genotyped. It's a part of their medical file. To require a companion diagnostic for every targeted therapy is probably not the right answer too. So there has to be a balance there. And FDA is starting to listen to that.
And data mining rather than doing another test makes sense.
Yeah.
Absolutely.
Absolutely. Terrific. Well, with that, let's please join me in giving our panel a warm applause. Thank you. And we're going to take a short, about 10-minute break before our next panel. Thank you.
I think it is clear how fortunate we are to believe in the power of ideas.
Supply the common sense and the fresh thinking of the Manhattan Institute.